Hi guys, hope this will find you in good health. Today we will be starting with the protein metabolism and we have series of lecture on this protein metabolism briefly. The notes are available already uh, on my websites. So today we will be in this initial lecture we will be talking about protein and the amino acids. So to understand the metabolic process of the protein, we have to understand about the chemistry of protein and amino acids. So as you know that the amino acids that are considered as building blocks for the proteins, and there are so many amino acids, there are about 20 amino acids that are being used to make different kinds of proteins. So let's start with this one. Uh, you know that the protein are made up of 20 different amino acids, and from nutrition point of view, there are 9 of amino acids that are considered as essential amino acids. What does it mean? Those 9 amino acids that cannot uh, generated in your body, and you have to take it from outside source through your diet. So these are called essential amino acid, and these are the 9 amino acids. So what is the composition chemistry of these amino acid? Each of the amino acid that comprises of one amino group, one acid group, one hydrogen, and a side group. Side group mean alkyl R. So it is a side group. The basic structure is the same, but it's a side group R. That means R that makes each amino acid unique. In the following slides, we will be looking at the structure. The sequence of amino acid in each protein that determine the unique shape and function of that. So look at this one. This is a typical amino acid. So as I told you earlier that each amino acid composed of one acid group. This is the acid group. This COOH. COOH. This is a acid group, right? COOH. This is the acid group, and this is the amino group. This is the amino group, NH2. You can see NH2. This is the amino group. This is the NH2. And there is the third thing that I have told you. With this carbon, hydrogen atom is attached. So up to this point, I have told you three things. One is the acid group. This is the acid group. This second number, this is the amino group, NH2. And third thing I have told you, that is the H. And as you know that the four component that can be attached with this carbon. So 1, 2, and 3. And the fourth one is the actually the R, alkyl group. This is the alkyl group, right? So that alkyl group actually varies. This is the alkyl group that varies with each amino acid. And this structure, amino group, acid group, and H, that remain the same for all the amino acid. Only thing that vary, that is the R group, right? So amino acid unique side groups. This I am talking about the R that result in difference in the size, shape, and electrical charge of the amino acid. And from the nutrition point of view, one of the classification I have told you that we can classify into essential and the non-essential amino acid. Essential amino acids are those that are required from our diet and our body cannot generate it. Non-essential are the amino acids that can be generated by our body. So non-essential amino acid include alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine, and the tyrosine. So these are the amino acids that are generated by our body, human body. So these are the structure. Glycine, you have to uh, memorize these kind of the structures. So look at this one. This is the actually the same. This is the same in all the amino acids. Only you have to remember this point, this point. That is in white portion, this point, this point, this point. So this is the same structure. One is the CWH, on the other side NH2, and on the third side H with this carbon, right? So this is the same structure, only you have to remember this part, this part. So for glycine, this part have H on it. For the alanine, this is the CH3. This is the same, this is the same, this is the same, and this it will be in alanine CH3. In aspartic acid, CH2 COOH. What is this? R part. This is the R part. This is will be the same. This will be CH2 COOH. And for phenylalanine, a benzene ring with CH2. A benzene ring with CH2, that will be. So in this white part, in this white part, you can see in this white part, this will be a variable. So essential amino acids, these are also called indispensable amino acid and must be supplied by the food people consume. What are these essential amino acids? Histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. So these are the amino acids that cannot be generated by the human body. So conditionally essential amino acids refer to amino acids that are normally non-essential but essential under certain conditions. Histidine is called as the conditionally essential amino acid. So this is the list of all the amino acids. Some of these are the essential amino acids and some of these are the essential list of the and these are the nutritionally non-essential. So you have to remember all of this essential and the non-essential amino acids. So now talking about the proteins, as we have talked about that the proteins that are made up of the amino acids. We have talked about the 20 amino acid essential and the non-essential. So all of that proteins are made up of that. So these amino acids that can join in different fashions. Maybe two amino acids are joined together to make up a peptide structure that will be called as dipeptide. If three amino acids are joined together, that is called as the tripeptide. And if more than two amino acid bonded together, that will be called as the polypeptide. So amino acid sequences all different, which allows for a wide variety of the possible sequences. So look at this one, how they join together to make a peptide bond. Remember, when two amino acids join together, that is joining by peptide linkages through peptide bonds. Look at this one. This These are the two amino acids. This is the amino acid 1. This is the amino acid 2. Look at this one. On one end, this is the central carbon that can be joined with 4 items. COOH on one side, on other side NH2, on other side NH2, and on third side H, and on the fourth side that I told you that may be variable in different amino acid, that is the R, right? So when this two amino acid join together, what happens? This is another amino acid, same structure, COOH on one side, NH2 on other side, and H on third side, and R on fourth side. So when two amino acid join together, COOH part of first amino acid, COOH part of first amino acid, and NH2 part of second amino acid that participate in peptide linkages. So what is happening? HO part of this amino acid, HO part, and this hydrogen from this amino acid that is used in this linkage. You know HO and H, that is making the water molecule. So you can see here, this is the water molecule. OH from first amino acid and this H from second amino acid, thus forming the water molecule. So when peptide, two amino acid link through the peptide linkages, there is release of water molecule, and when they are joining together, OH from first amino acid and H from second amino acid that participates, right? So that is making a dipeptide linkage. Why that is a dipeptide? Because this is a combination of two amino acid, two amino acid. So when it release OH from this one and H from this one, what is happening? CO and H. CO, what remained here? CO from this part, or and NH from this part. So look at this one. CO from this part, from first amino acid, and NH from this amino acid that join together. So what is the nature of this peptide link? Is CONH, CONH. So we write CONH. So this is the actually the peptide linkage, and we call it a dipeptide because two amino acids are linked together. So we conclude here our lecture today. Thank you very much.